Hello friends, welcome to Sam Biology class. Today we shall discuss about sex linkage and sex-linked inheritance. X and Y chromosomes can be divided into homologous regions and differential regions. Homologous regions contain DNA sequences that are substantially similar on both sex chromosomes, whereas the differential regions contain genes which have no corresponding allele on the other sex chromosome. Hence, in male, the genes in the differential region are said to be hemizygous. X chromosome contains many hundreds of genes, most of which are not involved in sexual function, and most of them have no corresponding allele on Y. Whereas Y chromosome, which is comparatively smaller than X, contains few dozen genes, some of which have corresponding allele on X and remaining do not have. The genes that are localized in the differential region of Y chromosome are involved in male sexual function. For example, gene SRY that determine maleness is localized in the differential region of Y chromosome. In addition, several other genes that are responsible for sperm production are also present in the differential region of Y chromosome. Genes that are localized in the differential region of X show an inheritance pattern called X-linkage. Similarly, genes that are localized in the differential region of Y chromosome exhibit Y-linkage. In general, genes that are localized in the differential region of X and Y chromosomes are said to show sex linkage. The inheritance pattern of sex-linked genes differ from the inheritance of autosomal genes. In case of autosomal inheritance, male and female progeny show inherited phenotypes in exactly the same proportion as typified by Mendel's result. But in case of sex-linked inheritance, male and female progenies exhibit different phenotypic ratios. In case of human being, X and Y chromosomes possess two homologous regions, one at each end. These homologous regions are autosomal-like, hence they are known as pseudo-autosomal region 1 and pseudo-autosomal region 2. One or both of these regions pair during meiosis and undergo crossing over. In this way, X and Y act as pair, and finally they are segregated into equal number of sperms. Now, what is sex-linked genes? Genes that are localized on sex chromosomes are known as sex-linked genes, which are of different types such as X-linked genes, Y-linked genes, and XY-linked genes. Genes that are present on the differential region of X chromosomes are known as X-linked genes, whereas those are localized in the differential region of Y are known as Y-linked gene or Hollandric gene. Finally, genes that are localized on the homologous region of X and Y chromosomes are known as XY linked genes. In addition, there are several other genes whose dominance is influenced by the sex of the bearer. Such genes are called as sex-influenced genes. Sex-influenced genes. Thus, male and female individuals may be similar for a particular trait but give different phenotypic expression of the same trait. For example, baldness. In men, baldness can occur due to disease, radiation, or thyroid defects. In some families, it is found to be an inherited trait. In such type of inherited baldness, hair gradually become thin on head top and ultimately leaving a fringe of hair low on the head. This type of baldness is known as pattern baldness. The gene for baldness B is found to be dominant in male but recessive in female. In heterozygous condition, the gene B expresses itself but only in the presence of male sex hormone. Three different genotypes are found where both the alleles are dominant, and another is heterozygous condition in which one allele is dominant and the other allele is recessive. In other case, both the alleles are recessive. So these are the three genotypes. The phenotypes are different in man and women. So here let us write down the phenotypes in man and in women. When both the alleles are dominant, baldness occurs in man and women. Baldness occurs in both man and women. But in case of heterozygous condition, baldness occurs in man, but non-bald condition occurs in women. In heterozygous condition, the gene is expressed only in the presence of male sex hormone, hence baldness occurs only in men, not in women. But when both the alleles are recessive, then non-bald condition arises in both men and women. Non-bald condition arise in both men and women. In addition to this, there are certain autosomal genes whose phenotypic expression is determined by the presence or absence of one of the sex hormones. The genes are known as sex-limited genes. Sex-limited genes. Their phenotypic effect is limited to one sex or the other. In other words, the penetrance of a sex-limited gene in one sex remains zero. The genes are mainly responsible for secondary sex characters in cattle, human beings, and fowl. For example, bad development. Bad development is a sex-limited trait in human being, as men normally have bad, whereas women do not have. Similarly, genes for male voice, body hair, and physique are autosomal in human being, but they are expressed only in the presence of androgens, which are absent in female. As previously mentioned, genes that are located on the differential region of sex chromosomes are known as sex-linked genes, and the characters determined by them are sex-linked characters, and their inheritance is referred to as sex-linked inheritance, which is different from the inheritance of characters determined by autosomal genes. Sex-linked characters are of two types, X-linked and Y-linked characters. A familiar example of sex-linked inheritance is red-green color blindness, which is a X-linked recessive character. The gene responsible for red-green color blindness is located on the differential region of X chromosome. Its dominant allele is responsible for the formation of color-sensitive cells known as cones in the retina of the eye. Cones are responsible for color vision. Its recessive allele is unable to produce color-sensitive cells. As a result, individuals become colorblind. Because females have a pair of X chromosomes and males have only one, so homozygous recessive females and hemizygous recessive males are colorblind. When normal vision female is married to a colorblind male, then all F1 offsprings are normal. F1 sons are normal because they receive their X chromosome from mother, which has the dominant allele. Daughters are normal but they are carriers. They are normal because they receive one X chromosome from mother, which has the dominant allele, and the other X chromosome from father, which has the recessive allele. So daughters are heterozygous. When F1 sons and daughters are inbred, then all the females are normal, out of which one is carrier. And regarding sons, one is normal and the other one is colorblind. So in this cross, normal and colorblind individuals are produced in the ratio of 3:1. Similarly, in another cross, when a colorblind female having two recessive alleles is married to a normal male, then in the F1 generation, all females are normal but carrier, but all sons are colorblind. All sons are colorblind because they receive the X chromosome with recessive allele from mother. So all F1 sons are colorblind. But the daughters are normal because they receive one X chromosome with dominant allele from father and the remaining X chromosome with recessive allele from mother. So here also daughters are heterozygous. But when F1 sons and daughters are inbred, then colorblind and normal individuals are produced in ratio of 1:1. So friends, this is all about sex linkage and sex-linked inheritance. For more updates, please subscribe my YouTube channel, Sam Biology Class.